Hello everyone, hope all are doing good. Well, in the previous class, I guess we discussed about the proteins. Well, in today's session, we'll be starting with a fresh concept under Unit 4, that is metabolism. So, we know that living organisms, they do have a complex internal order. So, if you consider a complex organism like human being, everything should be in its place, function efficiently at the right time. That is, everything should be equisitely organized or ordered. So to maintain the order that characterize the living state, living organisms must import food or energy from their surrounding. So there should be some mechanism which is involved in converting the food to the energy and the consumption of energy to bring about the processes that takes place inside a complex organism. So the complex coordinated network of enzymatic reactions carried out by living organisms to maintain the living state is referred as metabolism, or in simple words, you can easily define metabolism, a term for a set of chemical reactions that occur in the cells of living organisms to sustain life. And these metabolic processes that takes place inside the body, uh, it might be respiration, might be excretion, or might be any other phenomena or process. They lead to growth and reproduction and allow living organisms to maintain their structures and respond to the surrounding environment. So metabolism, it often involves a number of reactions, and these number of reactions are catalyzed by a sequence of enzymes. So such sequences of enzymatic reactions are referred as metabolic pathways. I shall repeat. Metabolism, it involves a number of reactions. Those reactions which are usually catalyzed by the enzymes which play their role in a sequential order. And this sequence of the reactions that are catalyzed by the enzymes are referred as metabolic pathways. And in the metabolic pathways, the product of one reaction will be playing as a substrate for the next reaction. The product of one reaction will play a role of substrate of the next reaction. And the successive intermediates that are formed in the metabolic pathways are referred as metabolites. That can be product of one reaction, or it may also act as substrate to the upcoming reaction. They are referred as metabolites. So there are many purposes for a metabolism, but let us consider the main purpose of metabolism. The first one is to convert the food to energy in order to run all kinds of cellular processes. Next, the conversion of food or the fuel to building blocks for proteins, lipids, nucleic acids, and some carbohydrates. And the third is the elimination of nitrogenous wastes. These are the three major purposes of a metabolism in the living organism. So in this unit, we are going to discuss about the metabolism and the definition of catabolism and anabolism along with examples. Then we have different metabolic pathways, under which we have carbohydrate metabolism. Under that, we'll be discussing about glycolysis as well as the citric acid cycle, which is also referred as Krebs cycle or TCA cycle. And we'll be discussing about the energy that are involved as well as the energy that are given out during the metabolic pathways. Then we'll be discussing about the gluconeogenesis. Again, it's a process of conversion of glucose to glycogen. And under fatty acid metabolism, we have beta-oxidation pathway. And under protein metabolism, we have urea cycle, and we have some other small concepts which are interconnected to those pathways. So we'll be discussing one by one. So to go with, hope you understood what is metabolism and what's the purpose of metabolism. So metabolism, it can be further distinguished into two phases. One is catabolism and the other is anabolism. So catabolism, as you can see on the screen, breakdown of larger molecules. What the catabolism does, it is a degradative phase of metabolism where the complex molecules are broken down into simpler ones, and these simpler small molecules are in turn taken up by the anabolism phase, which combines those smaller molecules to give rise to bigger molecules. So metabolism can be distinguished into two phases, catabolism and anabolism. Catabolism, it is the breakdown phase of metabolism, where you can find the complex molecules will be broken down into simpler ones. And during the breakdown of these complex molecules into smaller molecules, some amount of free energy will be liberated out. Okay. Then coming to the anabolism, it is a building up phase of metabolism where the smaller molecules that are obtained from the catabolism will be built up to form bigger molecules. And the main function of anabolism, it is to synthesize the larger molecules which are required for cell maintenance, growth, as well as the reproduction. And it requires both the free energy as well as the raw materials that are released by catabolism. So here catabolism is kind of giver and anabolism is a kind of taker, because catabolism, it breaks larger molecules into smaller molecules, and some amount of energy is released during this process. And the energy that is released during catabolism and the molecules that are broken down will be transferred to the anabolic phase, where the smaller molecules are built up to the larger molecules by making use of the energy that is given out during the catabolism in order to build or synthesize those molecules which are much required for the processes like growth, reproduction, as well as maintenance. So if I just like to compare between catabolism and anabolism, catabolism, as I said, it is a degradative phase, it's a breakdown phase, but your anabolism, it is a synthetic phase. Catabolism, the reaction is exorganic in the sense it gives out energy, but your anabolism, it is endergonic since it requires energy. And catabolic processes are oxidative in nature, while the anabolic processes are reductive in nature. Well, this is just a pictorial representation of how exactly catabolic as well as the anabolic processes that takes place within a cell. Here you can see you have larger molecules. Okay, so they go through this catabolic phase where they are made into smaller molecules like amino acids, nucleotides, etc. And there is liberation of ATP. ATP is nothing but adenosine triphosphate, which is a means of energy. And these ATP molecules as well as the smaller molecules, they in turn go to the anabolic phase where the smaller molecules are built up by consuming this ATP and they are made into the larger molecules. It is just to make sure that you understand. You did not draw this diagram. If you understood the exact meaning of anabolism and catabolism, that's more than enough. Well, as I said, we have many metabolic pathways. So to start with, we have carbohydrate metabolism. Carbohydrate metabolism is the whole of the biochemical processes responsible for the metabolic formation, breakdown, and interconversion of carbohydrates in living organisms. We know that carbohydrates are very important to cellular metabolism because carbohydrates are the primary products for the process like photosynthesis as well as starch. And we know that carbohydrates are the ultimate energy and they are the carbon source for most of the non-photosynthetic animals. As I said, carbohydrates are central to many essential metabolic pathways. And the most important carbohydrate is obviously your glucose, which is the most abundant organic molecule which is found in the biosphere, and it is the main metabolic fuel because it's a source of energy. Glucose can be converted into source of energy. Glucose is also central to our metabolism. It is one of the two major fuels in our bodies which do provide energy. One is the glucose and the other is your fatty acids. And your glucose, it is mainly obtained from the starch and obviously from the plant products, we know that. And these are digested, I mean the carbohydrates are digested to glucose and later it will be absorbed by the bloodstream. We know all these things. Glucose concentration in the blood is carefully controlled, ensuring that all parts of the body are constantly supplied with energy. I guess in one of our concepts we have discussed about the concentration of the glucose. What if the glucose concentration is high and what if the glucose concentration is low? And if you consider the importance of glucose, we know that most of our body tissues, they do require fatty acids as the alternative fuel, right? Because fatty acids are those tissues which in turn provide energy. But when we come to the brain doesn't work on fatty acids. We all know that brain depends only on glucose for its proper working. If the glucose level, it falls drastically below the range, that is, we have a range of 60 milligram per 100 ml. So if the human's blood glucose level, if it falls less than 60 milligram per 100 ml, then there are chances that the person may undergo coma or sometimes to death. So it's very important that a human's body has an average concentration of blood glucose. And whenever glucose is required, we know that the excess glucose will be converted to glycogen and it will be stored in muscles and in the liver because of the hormones like insulin and we have vasopressin. So whenever there is requirement of energy, so these glucose molecules are in turn converted to some other products through metabolic pathways. We have like glycolysis. It's a process of converting glucose into pyruvic acid, which in turn releases some ATP molecules. And we have citric acid cycle, that is TCA cycle. And we have one more, that is electron transport chain. So in coming classes, you will understand what exactly the glycolysis, citric acid cycle, and electron transport chain are. So carbohydrate metabolism, yeah, it is nothing but it's a series of biochemical processes which are responsible for the process that takes place in living organisms. And under carbohydrate metabolism, we have many metabolic pathways like glycolysis, TCA cycle, and as well as electron transport chain, which are in turn concerned with the release of energy in terms of ATPs. So this is carbohydrate metabolism. Here you can see you have glucose, then which is interconvertible with glucose 6-phosphate. So glucose 6-phosphate undergoes glycolysis, resulting in the formation of pyruvate, giving rise to 2 ATP energy molecules and 2 NADH. I shall explain what is NADH in the coming slide. And this is pentose phosphate pathway where the glucose 6-phosphate is converted into pentose phosphate. Again, there is a release of 2 NADPH. Again, it is one of the term concerned with the energy. Then you have gluconeogenesis where there is release of 6 ATP and 2 NADH, resulting in the formation of pyruvate. Then you have TCA cycle here, where 1 ATP, 3 NADH, and 1 FADH2 molecules are obtained. And here you have glycogenolysis. So these are the many pathways that are involved in the conversion of glucose into different products, which in turn involved in the release of energy. You should always remember the metabolic pathways are mainly meant to run the process in the body with the release of energy. So to go with, we shall go with the first metabolic pathway under carbohydrate metabolism. We have glycolysis. Glycolysis, it is one of the linear sequence of reactions by which the glucose compound, which is a 6-carbon compound, it is enzymatically, in the sense, in the presence of enzymes, is converted into 2 molecules of pyruvic acid. There is nothing but your pyruvate, which is a 3-carbon compound. So glycolysis, it's nothing but a reaction of linear sequence where a glucose molecule of 6-carbon atom is being converted into 2 molecules of pyruvate or pyruvic acid, which is a 3-carbon compound. And this metabolic pathway, I can consider this as metabolic pathway because you can see many products which in turn act as substrates to the next reactions, and there are many enzymes which act as catalysts to bring about this conversion of the reactions. And you can see the energetic, that is the ATPs and ADPs that are involved throughout this metabolic pathway. Considering glycolysis, it has 10 steps, and this process takes place inside the cytoplasm. And you can see none of the steps are reversible, so the entire metabolic pathway is irreversible. The glycolysis, it's a linear sequence of reactions where a glucose molecule, that is 6-carbon glucose molecule, is being converted into 2 molecules of pyruvate or the pyruvic acid, which is a 3-carbon atom. And this entire metabolic pathway takes place within the cytoplasm of any cell, that is of cells of any organism. And it is enzymatic sequence because you can see many enzymes are involved in the conversion of many products into substrates, and it is energized in the sense many ATPs as well as ADPs are involved in this process. In the sense, there is consumption of energy as well as release of energy during this process. So we shall go step by step. This reaction actually it is common to both aerobic and anaerobic respiration. And this glycolysis which is also referred as EMP pathway. Glycolysis process is also referred as EMP pathway, means Embden-Mayer of Paranas pathway, because the glycolysis mechanism was named after its discoverer, that is Embden-Mayer of Paranas pathway. As I said, this is common to both aerobic and anaerobic respiration. So it's very important to you people to know what is aerobic respiration as well as anaerobic respiration. Aerobic respiration is one kind of respiration which takes place in mitochondria, and this process it requires oxygen as well as glucose. It also produces some amount of energy, water molecule as well as carbon dioxide. Coming to the anaerobic respiration, this process it also produces some amount of energy, but lesser when considered to aerobic respiration, and it also requires glucose, but it doesn't require oxygen during the process. So as I said, in the glycolysis, the glucose is converted into two molecules of pyruvic acid, and along with the formation of some ATP molecules. So let me start this pathway. So we should go one by one. So the initial molecule is your glucose. 6C indicates 6 carbon. So glucose undergoes phosphorylation. Phosphorylation is a process of adding up of phosphate. So glucose undergoes phosphorylation in the presence of an enzyme, that is hexokinase. Structures that are written inside the blocks, they represent the enzyme. So the glucose, a 6 carbon atom, undergoes phosphorylation in the presence of an enzyme, that is hexokinase, and it is in turn converted into glucose 6-phosphate. Glucose is converted into glucose 6-phosphate in the presence of an enzyme hexokinase, and during this conversion, one ATP molecule is consumed. Okay, one ATP molecule is consumed in the presence of magnesium 2+ ion. Now this glucose 6-phosphate, again a 6 carbon atom, it isomerizes into fructose 6-phosphate in the presence of an enzyme referred as phosphoglucoisomerase. It is phosphoglucoisomerase. There is no energy molecule, that is ATP is not involved because it is isomerization process. This fructose 6-phosphate, again a 6 carbon atom, again isomerizes into fructose 1,6-diphosphate in the, sorry, it's not undergoing isomerization. Fructose 6-phosphate, it is further converted into fructose 1,6-diphosphate in the presence of an enzyme, that is phosphofructokinase. So this conversion, it involves a molecule of ATP in the presence of magnesium ion. So the product is fructose 1,6-diphosphate, a 6 carbon atom. And this fructose 1,6-diphosphate, what it does in the presence of the enzyme aldose, it cleaves into, it gets divided into two molecules, that is one dihydroxyacetone phosphate, we usually call it as DHAP, dihydroxyacetone phosphate, DHAP, and the other molecule is your 3-phosphoglyceraldehyde, that is 3-PGAL. So now this DHAP, that is dihydroxyacetone phosphate, which contains 3 carbon atom, because the fructose 1,6-diphosphate containing 6 carbon atom is being cleaved into two molecules each containing 3 carbon atoms. So this DHAP, that is dihydroxyacetone phosphate, it isomerizes into a molecule. It isomerizes into a molecule of 3 phosphoglyceraldehyde in the presence of an enzyme referred as triose phosphate isomerase. I shall repeat. The DHP, the cleavage product of fructose 1,6-diphosphate, it isomerizes into one molecule of phosphoglyceraldehyde, 3-phosphoglyceraldehyde in the presence of an enzyme referred as triose phosphate isomerase. So as a resulting, we have two molecules of 3-phosphoglyceraldehyde. Am I clear? So two molecules of 3-phosphoglyceraldehyde, now they are oxidized or they are hydrogenated into two molecules of 1,3-diphosphoglyceric acid, and this conversion of 3-phosphoglyceraldehyde into 1,3-diphosphoglyceric acid takes place in the presence of enzyme referred as glyceraldehyde dehydrogenase. And during this conversion, two molecules of NAD will be converted into two molecules of NADH2. I know you people might be worrying about what exactly it is NAD and NADH2. NAD, it's nothing but a coenzyme. The abbreviation of NAD is nicotinamide adenine dinucleotide, which is obviously found in all organisms. Again, it's a source of energy. And this NAD, that is nicotinamide adenine dinucleotide, it exists in two forms. One is the oxidized form and the other is reduced form. If it is NAD plus, then it is oxidized form, which is ready to accept proton in terms of hydrogen. If it is NADH, it is in reduced form and it is ready to donate a proton in terms of hydrogen. Hope you understood what is NAD and NADH2. So what's happening here? Two molecules of 3-phosphoglyceraldehyde, it is oxidized or hydrogenated into two molecules of 1,3-diphosphoglyceric acid, and in this then later this two molecules of 1,3-diphosphoglyceric acid are further converted into two molecules of 3-phosphoglyceric acid, and you can see in between the conversion of 3-PGL to 1,3-diphosphoglyceric acid, two NAD molecules are being converted into T2NADH2 molecule, that is the reduced form of NAD. Again, it's a source of energy. And 1,3-diphosphoglyceric acid in the presence of enzyme phosphoglycerokinase is converted into two molecules of 3-phosphoglyceric acid, and here the energy is being liberated. In the previous steps, you saw that energy was consumed, but here two molecules of ATPs have been liberated, and this process takes place in the presence of magnesium ion. Two molecules of 3-phosphoglyceric acid again are reduced to two molecules of 2-phosphoglyceraldehyde in the presence of enzyme referred as phosphoglyceromutase. And now these two molecules of phosphoglyceraldehyde, what they do, they lose a molecule of water in the presence of an enzyme referred as enolase to form two molecules of phosphoenol pyruvic acid, what we usually call as PEP. PEP molecule, that is nothing but your phosphoenol pyruvic acid. Now these two molecules of PEP, that is phosphoenol pyruvic acid, they undergo dephosphorylation. In the sense, they lose phosphate molecule. And again, you can see in this step, there is liberation of two molecules of ATP. And this conversion of 2-phosphoenol pyruvic acid PEP to 2 molecules of pyruvic acid. It involves enzyme referred as pyruvate kinase, and it is a dephosphorylation step where a phosphate group is being removed from the PEP molecule, and 2 molecules of ATPs have been released in the presence of magnesium ions. So this is how the glucose molecule is being converted into 2 molecules of pyruvic acid, and this entire metabolism, it occurs in the presence of enzymes, and this pathway, it involves both consumption of energy as well as liberation of energy. So to make this pathway simpler, I have written the entire pathway in reaction wise. Here you have, so here you can see glucose on consumption of ATP molecule in the presence of hexokinase and Mg2 plus results in the formation of glucose 6-phosphate. As I said, it is a phosphorylation step where a phosphate is being added to glucose, right? Then this glucose 6-phosphate again isomerizes in the presence of phosphate glucoisomerase into fructose 6-phosphate. Then the fructose 6-phosphate, upon consuming a ATP molecule in the presence of Mg2 plus and the enzyme phosphate fructokinase, results in the formation of fructose 1,6-diphosphate. Okay, then this fructose 1,6-diphosphate, now it is a 6-carbon atom, so it cleaves into DHAP. So what is DHAP? It's nothing but your dihydroxyacetone phosphate, and the other molecule, that is your 3-PGAL, that is 3-phosphoglyceraldehyde in the presence of an enzyme referred as aldose, right? Now this dihydroxyacetone phosphate, it isomerizes into a molecule of 3-PGAL in the presence of an enzyme referred as triose phosphate isomerase. So now you have totally 2 molecules of 3-phosphoglyceraldehyde, right? Yes, now these 2 molecules of 3-phosphoglyceraldehyde in the presence of NAD, that is your nicotinamide adenine dinucleotide, and phosphoric acid, they get converted into 2 molecule of 1,3-diphosphoglyceric acid, and here you can see there is conversion of NAD into NADH2, 2 molecules of NADH2. As I said, NADH2 is also a term involved with energy. I shall explain how it is involved in the next slide. Now this 1,3-diphosphoglyceric acid, they are in turn converted into 2 molecules of 3-phosphoglyceric aldehyde in the presence of phosphate glycerokinase, and here you can see 2 molecules of ATPs being liberated during the process. And these 2 molecules of 3-phosphoglyceraldehyde, they are then converted into 2 molecules of 2-phosphoglyceraldehyde in the presence of phosphate glyceromutase. And again, these 2 molecules of 2-phosphoglyceraldehyde in the presence of enzyme enolase and magnesium ions, they lose a molecule of water, resulting in the formation of 2 mole of 2-phosphoenol pyruvic acid, what we call it as PEP. And these 2 molecules of PEP again undergoes dephosphorylation, where there is removal of phosphate group from the PEP in the presence of an enzyme referred as pyruvic kinase, resulting in the formation of 2 molecules of pyruvic acid, and this last step is involved with liberation of 2 molecules of ATP. I hope I have made these things clear. But make a note, whenever they ask you to give the metabolism involving glycolysis, you should write the entire metabolism in terms of this linear sequence, but not in terms of this reaction. I have just written this reaction in order to make sure that you understand each sequence in a better way, right? Yes. Don't make this mistake of writing this entire reaction when they ask you for the glycolysis metabolism. Please do sit and practice this sequence of glycolysis. But if you go through this set of reactions, it becomes very easy for you to understand the entire glycolysis sequence. Coming to the significance of glycolysis. Whenever a glucose molecule undergoes degradation, yes, it is a degradation process where the glucose molecules are linearly or sequentially broken down into two molecules of pyruvic acid. So whenever a molecule of glucose undergoes glycolysis, it releases 8 ATP molecules. During glycolysis, a glucose molecule or single glucose molecule releases 8 ATP molecules. So let us count here. Now here ATPs are consumed. So it's not considered. Again here ATP is consumed, not considered. So here in this step you can see ATP is being released. So we got 2 ATPs. Then again the last step we got 2 more ATPs. So totally 4 ATPs. So where are the remaining 4 ATPs? Yes, as I said, NADH, it is concerned with energy liberation. A single NADH molecule can release 3 ATP molecules. One NADH molecule can release 3 ATP molecules. So here you have 2 NADH2. It is 2 NADH2. 2 NAD undergoes reduction to give you 2 NADH2. And one NADH molecule can liberate 3 ATP molecules on oxidation. So 2 NADH molecule in turn liberate 6 ATP molecules via oxidation. So from 2 NADH, I got 6 ATP molecules. 6 plus 2, 8. 8 plus 2, so 10. But I told you that 8 ATP molecules are obtained during this process. Yes, we have to think about the ATP molecules that have been lost during the process. So in the first step and the third step, you can see 2 ATP molecules have been consumed by the conversion. So the net gain is 8 ATP molecules. Hope I have made this clear. So here I'm winding today's session. So in the next session, we shall go with the next metabolic pathway, that is Krebs cycle and the electron transfer chain. Then we shall go with fatty acid metabolism as well as the protein metabolism. So till then, take care. Don't waste your time. Please sit and practice. This unit is very important where you have many pathways. You may get confused. So it's better that you sit and practice all the steps. Till then, take care. Bye-bye. Hello everyone, hope all are doing good. Well, in the previous class, we just started with a new concept, that is metabolism. And under this session, we discussed what is metabolism and what's the main purpose of metabolism in a human body, and the two phases of metabolism, that is catabolism and anabolism. We also discussed the many metabolism processes that are taking place in human body, and we discussed about the carbohydrate metabolism, where glucose is the main reactant which undergoes a series of reactions, that is a metabolic pathway. Pathway, which in turn results in the formation of two molecules of pyruvic acid. And this is how exactly the reaction takes place. As I said, the product of one reactant will act as a substrate to the next reactant. And coming to the energetics of this glycolysis, the entire glycolysis process releases about 8 ATP molecules of energy. And these are the concerned reactions of each steps. Well, in today's class, let us discuss the fate of pyruvate, that is your pyruvic acid. Pyruvate, which is formed by glycolysis, it has got 3 different fates. In the sense, the pyruvic acid or the pyruvate can be in turn converted to 3 different forms, which literally depends on the species as well as the metabolic conditions. And the first pathway or the first process is the conversion of the conversion of pyruvate to lactate, and second one is the breakdown of pyruvate to ethanol, and the third is oxidative decarboxylation of pyruvate to acetyl coenzyme A. So these are the 3 fates or the pathways of pyruvate. So let us discuss one by one. Conversion of pyruvate to lactate. The reason behind the conversion of pyruvate to lactate is that the pyruvate cannot be oxidized under anaerobic conditions. Pyruvate cannot be oxidized under anaerobic conditions, but pyruvate needs to be converted to some other form in order to provide energy. And the conversion of pyruvate to lactate, it takes place under such anaerobic conditions where there is a vigorous contracting of skeletal muscles. This vigorous contracting of skeletal muscle obviously requires ATPs, that is the energy. And this conversion of pyruvate to lactate is also referred as anaerobic glycolysis, since this conversion takes place in the anaerobic condition. As I said, pyruvate cannot be oxidized. And the second most thing is that NADH, the NADH that is formed in the glycolysis, it should be reoxidized to NAD+. The NADH that is released during the glycolysis should be deoxidized to NAD+, because this NAD+ has got a role to play in the glycolysis process. But here it's very tough for the NADH to get deoxidized. That's the reason the pyruvate undergoes conversion to lactate so that it can release NADH in form of its reoxidized form, that is NAD+, and during this process some amount of energy is also provided. So this conversion has got 2 vital functions. As I said, it regenerates NAD+ by using up NADH, and second, it produces ATP during very intense physical activity where ATP cannot be produced by aerobic processes like TCA cycle, that is your citric acid cycle, or the Krebs cycle, or ETC. So here you can see the reaction. The pyruvate in the presence of the enzyme lactate dehydrogenase gets converted to L-lactate, that is LEVO form of lactate, and this process is accompanied by the reoxidation of NADH enzyme to its oxidized form NAD+, and this entire process is accompanied by a free energy change of minus 25.1 kilojoule per mole. Well, this is about the second fate or the pathway of pyruvate, where it gets converted to ethanol or broken down to ethanol. This process is also referred as alcoholic fermentation. And coming to this type of process, it usually takes place in case of microorganisms like yeast, which are much known for their alcoholic fermentation. And this process takes place in 2 steps. One, the pyruvate in the presence of an enzyme pyruvate decarboxylase, which is accompanied by an enzyme that is thiamine pyrophosphate. TPP is nothing but a coenzyme, thiamine pyrophosphate. With the removal of carbon dioxide molecule, pyruvate gets converted into acetaldehyde, and this acetaldehyde gets converted to ethanol, and this conversion is catalyzed by an enzyme alcohol dehydrogenase. Here also you can see NADH gets reoxidized to its oxidized form NAD+. If I talk about this ethanol that is produced as a breakdown of pyruvate in case of yeast, it is considered to be the waste product of yeast. Ironically, this excretory product of yeast, it is in great demand by a large chunk of humanity. Well, we have the third pathway or the third fate of pyruvate, where it undergoes oxidative decarboxylation, resulting in the formation of acetyl coenzyme A. This fate is very important since pyruvate once gets converted to acetyl coenzyme A. This acetyl coenzyme, it is an uh, uh, initial reactor in the case of the next metabolic cycle, that is your Krebs cycle. If I just consider glycolysis or the amount of energy that is discharged by the glycolysis, it amounts for only 7% of the energy that is present in glucose. If I consider the remaining metabolic processes such as citric acid cycle, the electron transport chain, as well as the oxidative phosphorylation, they amount for the largest amount of energy. In the sense, these processes release much more energy. But the thing is here, pyruvate itself can't enter the mitochondrial matrix where the exact Krebs cycle starts. Before the pyruvate can enter into the citric acid cycle, it has to be first converted to the acetyl coenzyme A. And this conversion is accompanied by a integrated multi-enzyme complex referred as pyruvate dehydrogenase complex, which is a combination of three important enzymes, E1 plus E2 plus E3. As well as this decarboxylation process is also accompanied by 5 different coenzymes. Here you can see the coenzyme A and your NAD plus, that is nicotinamide adenine dinucleotide in its oxidized form. Then you have your TPP, that is thiamine pyrophosphate. Then you have FAD, that is your flavin adenine dinucleotide, and your lipoic acid, also referred as lipo-A. And here NAD plus gets reduced to NADH, and this conversion, that is pyruvate, you can see here the carbon dioxide molecule gets removed, and this position is accompanied by the acetyl group of coenzyme A, resulting in the formation of acetyl coenzyme A with the removal of carbon dioxide molecule. So this is how the acetyl coenzyme is obtained from the pyruvate by undergoing oxidative decarboxylation, and this acetyl coenzyme A acts as initiator in your Krebs cycle, which is our next concept. Yeah, here we have the Krebs cycle. As I said, the pyruvate itself can't enter the mitochondrial matrix where exactly the Krebs cycle starts. It has to be converted to acetyl coenzyme A, and this conversion is brought about by the enzyme, that is pyruvic acid dehydrogenase, which is accompanied by an integrated enzyme complex as well as the 5 coenzymes. This process is referred as oxidative decarboxylation, and here we got acetyl coenzyme A with 2 carbon atoms in its compound. And this acetyl coenzyme A, which gains entry into the mitochondria, actually transfers its acetyl 2 carbon group to the oxaloacetic acid. What the acetyl coenzyme does, once it enters the mitochondrial matrix, it transfers its acetyl group to the oxaloacetic acid, which is already present in the mitochondria, and this transfer takes place in the presence of water, resulting in the formation of a 6-carbon carboxylic acid referred as citric acid, and this conversion is brought about by the enzyme citrate synthetase. This is your citrate synthetase. And here you can see a coenzyme molecule is released during this process, which later goes and attaches to the another pyruvic acid. I shall repeat again. So you get acetyl coenzyme A from the pyruvic acid via oxidative decarboxylation process, and this acetyl coenzyme A, once it gains entry into the mitochondrial matrix, it transfers its acetyl group to the oxaloacetic acid, which is already present in the mitochondria in the presence of the water, and an enzyme citrate synthetase results in the formation of a 6-carbon carboxylic acid referred as citric acid, and during this process a coenzyme A is released, which in turn goes and attaches to some other pyruvic acid to bring about next process. Now this citric acid, 6-carbon carboxylic acid, it upon losing a water molecule results in the formation of cis-aconitic acid. Since it loses a water molecule, it can be considered as a dehydrogenation process, and this takes place in the presence of an enzyme referred as aconitase. This is aconitic acid, again, which is 6-carbon carboxylic acid. In the presence of same enzyme, that is aconitase, it gains a water molecule. See here, the arrow mark is directed, the inward arrow, and this is the outward arrow. The outward arrow always indicates the removal, and the inward arrow indicates the addition of water. So this is aconitic acid, and in the presence of the same enzyme, that is aconitase, it gains a water molecule, resulting in the formation of isocitric acid, which is an isomer of citric acid. Again, this 6-carbon isocitric acid, it undergoes oxidation or dehydrogenation under the influence of an enzyme named isocitric dehydrogenase to form oxalosuccinic acid, which is again a 6-carbon compound. And here you can see, as I said, it is an oxidation or dehydrogenation process. So a hydrogen molecule will be removed in the form of H+, that is your proton, and that proton will be gained by the NAD enzyme, which gets reduced to NADH2. The so formed oxalosuccinic acid, the 6-carbon compound, it undergoes decarboxylation. Can you observe here the outward arrow indicating removal of carbon dioxide? So the process referred as decarboxylation, and this decarboxylation takes place in the presence of an enzyme oxalosuccinic acid decarboxylase, resulting in the formation of alpha ketoglutaric acid, which is a 5-carbon compound. Yeah, obviously carbon is removed in the form of carbon dioxide, so the number of carbon gets reduced to 5 number. This alpha ketoglutaric acid, again it undergoes decarboxylation. Here you can see the outward arrow where a carbon dioxide has been removed. So the alpha ketoglutaric acid undergoes decarboxylation to form succinyl coenzyme A, and this decarboxylation is brought about by the enzyme, that is alpha ketoglutaric dehydrogenase, and this decarboxylation involves the coenzymes like coenzyme A and NAD. Next, we have succinyl coenzyme A. This succinyl coenzyme A, the 4-carbon compound, undergoes a series of complex reaction, and this series of complex reaction take place in the presence of inorganic phosphate, ultimately giving rise to succinic acid, which is again a 4-carbon carboxylic acid. And during this process, a coenzyme A will be released. During this process, a coenzyme A will be released, which is released along with energy referred as GTP. In the previous metabolism, we used to talk about the energy term as ATP, but in this cycle, the energy is released in terms of GTP, that is your guanosine triphosphate. And you should remember this is the only reaction or the metabolism where GTP is synthesized instead of ATP. I should repeat. Succinate coenzyme, it undergoes a series of reactions, and these reactions are catalyzed by an enzyme that is succinic thiokinase, and this reaction takes place in the presence of inorganic phosphate, ultimately resulting in the formation of succinic acid. And during this process, a coenzyme A will be released along with some amount of energy, and that energy will be in terms of GTP, not in terms of ATP. And this is the only process where energy is released in terms of GTP. Now we have this succinic acid, a 4-carbon carboxylic acid. It undergoes dehydrogenation, and this dehydrogenation is brought about by the enzyme succinic acid dehydrogenase, or simply succinic dehydrogenase. As I said, it is a dehydrogenation process. Obviously, hydrogen will be removed in the form of proton, that is H+, which will be accepted by your coenzyme A, that is FAD, which gets reduced to FADH2. So succinic acid, in the presence of succinic acid dehydrogenase, undergoes dehydrogenation, where the coenzyme FAD will be reduced to FADH2, resulting in the formation of fumaric acid, 4-carbon carboxylic acid. Now this fumaric acid, here you can see the arrow is inward. So the water molecule is being added during the process. So upon addition, the fumaric acid in the presence of enzyme fumarase gets converted to malic acid. So it is a hydrogenation process. Now this malic acid, that is a 4-carbon compound, gets dehydrogenated in the presence of an enzyme malic dehydrogenase. So it is a dehydrogenase process where hydrogen will be removed in the term of H+ or proton, and that proton will be accepted by the coenzyme NAD, which in turn gets reduced to NADH2. Thus, malic acid gets converted to oxaloacetic acid, a 4-carbon carboxylic acid. So here, with the regeneration of oxaloacetic acid, the Krebs cycle is completed. Again, this oxaloacetic, which is already found in the mitochondria, will accept acetyl group from the acetyl coenzyme A, and in the presence of water, it gets converted to citric acid. This is how the cycle keeps on continuing in the mitochondrial matrix. And this is about the set of reactions. So it becomes easy for you to understand. So before starting with the exact cycle, go through these reactions. Then you can start drawing the Krebs cycle. And please, you need to practice this Krebs cycle, since the Krebs cycle is a bit complicated when compared to your glycolysis. though the number of steps remains same. In case of your glycolysis, it is a linear chain, but in case of your Krebs cycle, it is a bit complicated where the number of the products as well as the substrates are many, and even you come across many coenzymes as well as the enzymes which are reacting within the cycle. So please do sit and practice, and as I said before, practicing this entire complicated cycle, go through these reactions, which becomes very easy for you people to follow up that Krebs cycle. Coming to the significance of Krebs cycle, as per the Krebs cycle, the net gain of ATP during aerobic respiration, as I said, it is aerobic respiration. Whenever a molecule of glucose undergoes degradation, in case of Krebs cycle, 38 ATP molecules are liberated. I shall repeat, the net gain of ATP during the Krebs cycle, that is whenever whenever a single glucose molecule undergoes degradation, it results in the release of 38 ATP molecules. So the number of ATP molecules that are directly synthesized are 2 ATP molecules. Because this guanosine GTP can later be converted into 2 ATP molecules. And as I said, a single NADH2 molecule can give 3 ATP molecules. Hope you still remember when we are discussing in glycolysis, a NADH2 molecule can in turn release 3 ATP molecules. If you simply count in the Krebs cycle, we have under 8 NADH2 molecules. So obviously 8 into 3, whenever this 8 NADH2 molecule undergo oxidation, they release 24 ATP molecules. Then we have FADH2 molecules. There are 2 FADH2 molecules which are involved within this Krebs cycle, and one FADH2 molecule can liberate 2 ATP molecules when they undergo oxidation. Please remember this properly. GTP is equal to 2 ATP molecules, and one NADH2 molecule can in turn give rise to 3 ATP molecules. So therefore, there are 8 NADH2 molecules which are involved or which are liberated during the Krebs cycle. Therefore, when these 8 NH2 molecules undergo oxidation, they do release 24 ATP molecules. Then we have 2 FADH2 molecules, and each FADH2 can in turn give 2 ATP molecules. Therefore, when these 2 FADH2 undergo oxidation, they give out 4 ATP molecules. So totally 2 from GTP, 24 from 8 NADH2, 4 from 2 FADH2. So totally I have 30 ATP molecules. And I have to take account of glycolysis because we are taking pyruvate from the glycolysis process, isn't it? The pyruvate is considered to be a product of glycolysis. So we have to take account of the ATPs that are released during glycolysis. And we know the net gain ATP from glycolysis is 8. Therefore, the total ATP molecules that are gained via Krebs cycle is equal to 38 ATP molecules. Hope I have made this concept clear. Well, in the next session, we shall discuss the next metabolic cycle under fatty acid metabolism. I hope you have received the assignments, what I have made. Please sit and answer them and send me in time. Till then, take care. Bye-bye. See you in the next session.